Hi, everyone, and welcome to this edition of Budding Solutions, a series on disease and pest management in cannabis sativa. My name is Kim Leonberger, and I'm an Extension Associate in the Plant Pathology Department at the University of Kentucky. In this episode, we're going to take a closer look at damping off, a disease which can run rampant in greenhouse and field production. To discuss this important disease issue, I would like to introduce Dr. Nicole Gauthier. Dr. Gauthier is a professor and extension specialist at the University of Kentucky. Her program focuses on disease management of specialty crops, including fruits, vegetables, and hemp. In her extension role, she develops educational and outreach programs to help growers manage disease through identification, understanding of pathosystems, and integration of management strategies. Her research program focuses on a range of hemp diseases, including Fusarium head blight, as well as stand establishment and seedborne transmission. She's co-editor of the new APS Compendium for Cannabis Diseases. Dr. Gauthier's resources can be found at plantpathology.ca.uky.edu/extension/publications, on Facebook and Twitter at KY Plant Disease, and on YouTube at Nicole Gauthier. We'll put all of those links in the description for this video below so that you can access them easily. Welcome, Nicole. I know you've prepared a few slides to share with us about this disease, and then we'll have some time for a few questions.
Okay, so I'm going to share my screen. All right, so today we're talking about damping off diseases. So we're using the term damping off as more of a blanket term. So what is damping off? In general, it's a seed or seedling disease. In general, we focus on damping off on those seeds and seedlings, but those same diseases can also occur on cuttings. So whether it's non-rooted or rooted cuttings. But today we're going to talk about it as a seed or seedling disease. But just remember that in general, it kind of covers the gamut. So damping off diseases can be caused by fungi, or as we call them in plant pathology, true fungi, or water molds. So water molds are fungus-like, but not true fungi. And that's going to matter in the way we manage them. Damping off diseases really result in poor stand establishment. This can occur in greenhouses, indoor grow, or in open fields. In most of the continental US, these are early season diseases, usually when the soil is cool, when things are wet, if we have a cool, rainy spring, but they can occur anytime. In terms of hemp, in the field, we usually plant early mid-spring. But in greenhouses, generally it's warmer, so the dynamics may be a little bit different. So there are two types of damping off as we talk about them, as we describe them. The pre-emergent damping off is generally when we talk about seeds, whether the seeds fail to emerge, the seed may rot in the ground. As it starts to break dormancy, it will rot in the ground so it never comes up above that soil line. And pre-emergent damping off can be caused by a seedborne disease. So if the seed is infected before it's planted, or a soilborne disease, and that's when the pathogen is in the soil and the pathogen will attack the seed. The second type of damping off is post-emergent damping off. So that will generally occur when a seedling is infected after it germinates, so after it rises above the soil surface. So symptoms generally will be wilting or collapse of small plants at the soil line or below the soil line. So sometimes we'll see infection of the roots, sometimes of that crown, and sometimes we'll generally just see wilting and then overall seedling death. The complex thing about damping off, so in this series we've been talking about a single pest or a single pathogen, but damping off again is a wide range of pathogens, so we're collectively calling them damping off. The soil-borne pathogens can also be seedborne pathogens in some cases, can be caused by the true fungi, Fusarium and Rhizoctonia. By the way, this is not the Fusarium, Fusarium oxysporum that causes Fusarium wilt. This is generally going to be Fusarium solani, which is a soil-borne pathogen that will affect seedlings, roots, crowns. And the second group of pathogens, as I alluded to earlier, are the water molds. They're fungus-like, and these are the Pythiums. We know of about 5 Pythium species that can affect Cannabis sativa, and Phytophthora. Phytophthora, 1 or 2 species, depending on where you're located. In general, soil-borne pathogens, they live in the soil, right? So that hence the name. They move any way that soil is moved. So they can travel or be transported by water runoff, contaminated seeds, whether they're infecting or hitchhiking, improper greenhouse sanitation, any way that we move soil. So that can be tools, that can be equipment, that can be the bottoms of our shoes or dirty overalls. Disease will usually develop in wetter soils, especially those water molds, but wet soils. Most of the time when that soil is a little bit cooler, so earlier in the season, with the exception of one of the Pythiums, Pythium phanodermatum, which likes hotter temperatures. But in general, it's cool, wet soil where we're going to see most damping off disease. These pathogens, along with a few others that I'm not going to go into detail today, but they generally have very wide host ranges. So they can also be infecting weeds, previous crops if you're on a crop rotation. Generally, these pathogens will survive on crop residues, any crop that's a crop or crop parts that are left in the ground. Again, weeds and overwintering. That's a very important component. Overwintering on debris and by propagules. So some of them have overwintering structures or oversummering structures where they will survive from season to season. So we manage soil-borne diseases primarily by cultural management. So we're going to start by using clean seeds, clean cuttings, so as not to introduce these pathogens into our fields or indoor production facilities. And definitely so we get a good start, right? Well-drained soils or well-drained media is critical. Again, a lot of these pathogens like the wetter soil conditions. If you're in the field, raised beds, any kind of surface drainage and internal drainage, very important. Waiting until the soils are warmer, so timing that crop, and this is especially true for field production. And then in between crops and in between working in the field or in between different blocks or zones or across a larger farm, disinfesting tools and equipment in between each of these times that you're working. So not entering a field with dirty equipment from a previous block. So remember, those pathogens will hitchhike. If you're in a greenhouse, we're adding a few more management strategies. Of course, we're managing water in the greenhouse, and that can be everything from wet floors to dirty hoses. Always using foot baths, keeping things as clean as possible. So just because it's a greenhouse or indoor grow doesn't mean it can be dirty. Fallowing your greenhouse or indoor production facility between crops and sanitizing between crops. So not overlapping these crops. And every time we're starting a new crop, using clean, sterile soil, not reusing any media, and preferably either sterilizing containers if you're a small production or using new containers so as not to have that hitchhiking or that transfer of pathogens. In terms of management, we don't have conventional products at this point to manage our damping off diseases, but some biological fungicides that have potential can be the bacillus group. There's a lot of bacillus products, by the way. I'm lumping them together here. Clonostachys, Gliocladium, Streptomyces. There's a couple of different Streptomyces products. So these are some potential products. We just don't have the efficacy to be able to put solid recommendations together. So remember that without efficacy data, we're basing some of these general recommendations on what we know about these pathogens from other crops like vegetables and ornamentals. Biological pesticides work as preventative, so we should always begin preventative programs. Don't wait until disease pops up, and then combine these with those cultural practices that I discussed in the last slide. Remember too that biological pesticides, biofungicides are strain dependent. So a bacillus is not a bacillus is not a bacillus. So take that into consideration. And then, of course, the efficacy will depend on that type of pathogen, right? So is it which of the Pythium species, which of the Fusarium species? So that will make a really big difference. So in closing, Kim, some of our resources that I highly recommend is always to use a diagnostic lab. You cannot guess on which pathogen you have based on symptoms. The closest diagnostic lab to you, sometimes those are private. They usually are associated with your university. Get to know your local extension office. They're an excellent resource. Check out your university's resources. There's usually a lot online or a nearby university. That being said, here at the University of Kentucky, we have a hemp website where we're pulling in lots of different resources, hemp.ca.uky.edu. And then we have a multi-state group where lots of hemp pathologists and entomologists working together. And we are building a website right now, and that's through Cornell. So blogs.cornell.edu / S1084hemp. And we will put that link down in the description. And finally, there's not a lot of information on some of these products specifically for hemp or for cannabis sativa, but you can always look up that information of those pathogens from reliable sources. For example, looking up Pythium in general on other crops might lead you to some understanding. So with that, Kim, I will stop sharing my screen and take any questions.
Wonderful. Thank you so much for sharing all of that information with us. It was very valuable. So just a couple of questions for you. One, you had mentioned that there are multiple pathogens that can cause, in general, damping off. So how does a grower know which one they have, or does a grower need to know which one they have?
Grower needs to know which one they have, but a grower typically will not know. So it's really important, like I said, to get to know your local extension office. They can guide in where to find the nearest and most reliable diagnostic lab. So a diagnostic lab will tell you which pathogens you have. Now, of course, after that, you should have a history of disease on your site. So maybe every year, if you have a damping off problem, you would get that information. But in time, a grower will know. And based on what they know from a previous crop, they can use preventative action in the future.
Wonderful. And then kind of also following along that same line of there being different pathogens, do you see certain ones more frequently in indoor production versus, field or outdoor production, or is it really kind of the same group of pathogens all the way across the board?
So in indoor production, typically those are the Pythiums. I mentioned that Pythium aphrodermatum is the most common of the greenhouse Pythium species, but that doesn't mean that's what you have. So don't just take that as a definitive. But usually the Pythiums are the most common in indoor production. So that could be indoor and greenhouse. In the field, though, that's really variable, and it often depends on the history of the crops in those fields. So the pathogens that I talked about today, the damping off pathogens, generally have a very wide host range. So they can be affecting crops before. So a year, 2 years, even 3 years before you putting that cannabis crop into that field. And that could be agronomic crops, horticultural crops, forages, weeds. So not to take that, so knowing a history of a field can give you some guesses, but using that diagnostic lab, it's not that expensive. And trust me, a $25 or a $50 diagnostic fee is pennies compared to the cost that would be affiliated with losing large amounts of your crop, especially at that very early stage.
Excellent. One last question for you. It seems like there's a lot of room for there to be new knowledge gained in this area, that there's still a lot of room for research that could happen in this space. Would you be willing to talk about any new or ongoing research that you know about that it's related to damping off?
Yeah, so Again, a lot of the research that's ongoing right now is in the hemp space, and a lot of that is just tied to funding. And so our group in particular are working on what we call stand establishment. So in field hemp, stand establishment has been a problem for quite some time. So we're trying to get to the bottom of it. And then the pathology component of our larger project, we're looking at which soil-borne pathogens are there in our field, which ones are infecting seeds, which ones are infecting seedlings. And then, of course, the big question is how many of them are seedborne? Or clean seeds coming in, or clean seeds being planted and then becoming infected? So that is where our focus is right now. And hopefully, this year we'll be able to get more answers and be able to share that with our audiences.
Excellent. That sounds really exciting. So it sounds like you have a really exciting year ahead of you. Well, thank you again so much for joining us. I really appreciate you taking the time to share all of this valuable information with us. So just a reminder to our viewers, please make sure to check the description for this episode for all of these valuable resources that we've talked about. The links will be posted there, and we look forward to seeing you again on another episode.